Okay, I'll be giving this lecture again because I forgot to record this. So what I'll be doing today is I will be recording the lecture on my computer and then uploading it. Instead of a Zoom lecture, you will be watching a recorded lecture, you know, by me. So that's why this will be a little bit more different. I hope this will be better in quality because there were some complaints that my connection was not really stable today. Okay, so yeah, this lecture will be about me. So what you have seen me since the beginning of the semester, but what do I do for research? So I will be talking about that, and I'll be talking about what I do in general. So, as Dr. Jensen pointed out before, I do a lot of research behind the microscope because I would say like some part of my research is collecting flies, bring them to lab, and measure their phenotypes, or like collecting them to send to sequencing or anything. So there is a part of my research which is in the laboratory. And you see here is a carbon dioxide pad where I knock the flies down. Here are some flies inside this tube, and the tube's bottom is like open, so they just there's a mesh over here, so they just pass out. Yeah, but am I behind the microscope all the time? Actually, I'm not, because a lot of my research is, like, I would say more than half of it is outside. I collect flies from the wild, bring them to lab. I do a lot of things in the lab, true, but then I create experiments, and then for some of the experiments I go outside and do the experiments outside. So let's start with how I collect flies. But before that, what we need to understand is what's a fruit fly. So the fruit fly, Drosophila melanogaster, is the species I'm working with. So here you see they're resting under the tree because they hide from the sunlight during the day, and I know all of these are Drosophila melanogaster because I've taken this photo from an experiment I set up in the orchard, in the experimental orchard we have. So the second question is, we call them fruit flies, but is it a really good naming? So because we collect them from the fruits, but they actually eat yeast. So they don't consume the fruit itself, but they consume the yeast growing on rotting fruit. So they are not really fruit flies, but they're kind of vinegar flies. They really like the smell of vinegar. They will, like, if you are trying to trap them at home, you can use vinegar to trap them, and that's why they're not really fruit flies. There are real fruit flies in the in all Diptera. The fruit flies normally eat fruit itself, the fresh fruit, and they eat the fruit. That's why they're real fruit flies. But the melanogaster, even though they're called fruit flies, they don't. So how do I collect them? So here we see an aspirator. Here's the mouthpiece. Here's the tip. I collect the flies with this aspirator. Dr. Jensen has shown an aspirator before. These are like more modern versions of that, where like we use these plastic vials, and this is like this is kind of, this is not very beaten up, you can say. Here you see a trap. So this trap has a small hole here. The flies go in from that hole. I generally put a bait inside, and when they try to go up, they cannot because they get trapped. But where do we find them, right? So like this is how we collect them in an orchard, like in an like in a farm, let's say. I hang these traps from the trees, and flies go in. But if it rains, the rain cannot enter because they're sideways. And here you see Paul. Paul is my advisor, and we are in the woods, like trying to go behind an orchard in Maine to get some flies from the apple press. So Noah is one of the friends I've made during my trips. He was really interested in my work. I was going to their farm to collect flies, and here you see him with a net. Netic is another technique that we collect flies. If there are a lot of flies, you can use the net to collect flies. So back to lab. So after collecting flies, I bring them back to lab, and I release them into these cages. You see there are a lot of them. And then after releasing them into cages, what I do is I sort them out. Like I basically collect them with the aspirator you see from the cage, and put them onto the CO2 pad where they fall asleep, and sort their species. So the reason is there are several reasons we do this. The first reason is that I need isofemale lines. Isofemale lines, you bring them to lab, you create a line where you control the genetics. So you know the mom of the flies, but you don't like there's one mother and multiple fathers because they made it before you collect them in outside. The second reason is that when we go collect flies and bring them to lab, we don't know if they are Drosophila melanogaster or Drosophila simulans. So their females are exactly the same, but their males differ a little bit. So like they only the penises of the males are a little bit different. The shapes are different, so we can tell the species from that. So that's why we create these isofemale lines. We put one female into one vial. It's like a tube with food. We put them into one female into one vial. They lay their eggs. The eggs hatch. They just develop, become adults, and then we look at their males, and we know what species they are. Then we sort them, and then we create all these female lines. Like we create thousands of isofemale lines in the lab, and then we use them to design experiments. So when I say experiments, we have this experimental orchard where we have like these 6x6x6 cages where there's a peach tree inside, a ground cover like a soil ground cover, and it's completely sealed off, so the flies cannot get out or get in. So we know what's going on. We increase their sizes up to millions in these cages. The other thing we use is for indoors experiments. So indoor experiments, you see, like I create these cages, and we do a lot of experiments indoors too. And when we are doing this, like we brought the isofemale lines. So isofemale, when you create isofemale line, you decrease the genetic diversity in that line by a lot, right? It's basically kind of inbreeding. when you're creating the experiment, you outbreed them again. So what you do is, let's say you have 100 lines, you put 100 lines together and say, okay, this will make my population, and you put them together, let them mate and increase in sizes. So you outbreed them again. This way we can control the amount of genetic diversity inside the population we created, so it is a good population to do experiments. All right. Okay, hunting flies is cool, but is it the science, right? Like, what is the science behind this? So I call myself an evolutionary ecologist. So I just, these are just some cool pictures and stuff. Actually, like, there's something wrong in this, but it's okay. I also put T-Rex here because it's cool. Everybody likes T-Rex. That's why. But yeah, these are just cool stuff. But it's not really what I do. What I'm trying to say. But I'm an evolutionary ecologist. So what I try to do is to answer the questions of ecology or like evolution using both. And what I do is I like the way I concentrate onto the questions is I try to answer evolutionary questions in ecologically relevant setting. What does this mean? So evolution in the lab is different than evolution in the nature. So that's what I'm trying to find out. What's happening in what is happening in the nature, and how does evolution work in the nature. All right. And then there's global climate change. Global climate change, we know it's happening. The ice is melting in Arctic and Antarctic. It is affecting all of our livelihood. We know the temperatures have been rising faster than ever in the last, I think, 20-25 years, and it will keep happening. But what is the effect? So what is the effect of this, right? I want you to watch this short video before I continue, which is about the sixth mass extinction. Okay, so climate change is doing a lot of things, but how it does it, like how, what's the relevance to this lecture, right? So let's consider wood thrush. If you know it, you know, it's a bird species in North America. It's a really cute bird species, by the way. But their ranges are contracting like a lot. Like they lost 57% of their range, and their vulnerability status is high right now. So what I'm trying to understand is, like, how can we reverse this, or can we, in at least in some of the species? So I'm interested. I'm trying to make the connection with my research and species ranges. Another thing is evolutionary rescue. Evolutionary rescue is, let's consider there is an environmental change at one point, and this environmental change, with this environmental change, the genetic structure of the population becomes like disadvantageous, and they start to decrease in numbers, and they start going like extinct. Something happens here which ends up in rescue. But what is the rescue, right? So the rescue is basically the genetic change that's happening. It might be a mutation. It might be a mutation you bring from another source, but like it is a small mutation that's introduced here, and it starts to increase in the population very fast because it is advantageous under this situation, and it increases by a lot. So the population, even though it was decreasing in the first phase, in the second phase it starts to recover, and in the third phase it starts going up again. So this is the abundance, this is the adaptive allele frequency as it goes down up. And you know, these are arbitrary, like it don't think these are as absolute values too. So okay, like, but how can we address this question? So I want you to concentrate on the question of how, but not actually what. Like, what I did is not really important, but how I did is kind of important. So I'll be talking about the effects of migration on rapid adaptation. So I worked on on Drosophila melanogaster to understand the effects of migration on rapid adaptation. So I will be connecting this migration on rapid adaptation to the range problem. The first thing we have to understand is the way I use the migration word is not equal to gene flow, even though it is in the literature they are used like interchangeably. Here I am making a big distinction. Migration, according to me, is the movement of the individuals, and gene flow is the resulting effect of migration, where you start seeing these alleles coming from the outside source in the population in some abundance. It doesn't have to increase, it doesn't have to decrease over time. Like, you know, if you see those alleles which were not there, or some effects of the migration in the population, then there is some gene flow. So like seeing the effect of the coming alleles in the population is gene flow. But I am not directly looking at the gene flow, I am looking at migration. So here, this is a illustration I just, you know, put together here I created the this is the population structure. As the population structure increases, the genetic diversity decreases, and it's kind of important because there are there are five major evolutionary forces we have to consider. Those are mutation, selection, drift, gene flow, and habitat preference. Two of these forces increase the population structure by increasing the variation in the allele pool, and those are gene flow and mutations. However, mutations are like for mutations, you're inventing something. If you're talking about a mutation, you are talking about inventing something new. But in gene flow, you don't have to invent anything new. You already have it. Like you have the gene somewhere else in some population. That it's just that gene comes to the like resident population. So in terms of that, gene flow is really important. So from literature, what do we know? So what we know, the first thing we know is the population, the fly populations in Americas. I will concentrate mostly in North America, are really new. They are like around 400 years old, and they came to the Americas with the colonialists because Drosophila melanogaster is a human commensal species, so they hang around with humans. We know that from not only from this introduction, we know that from this Africa to Europe introduction too, which happened if I'm not mistaken, it happened between a million years and 100,000 years or something. But it's not really important. It happened twice. So humans went to Africa to Europe, and Drosophila melanogaster did too. And they like, I think when they were passing Bering Strait, it was too cold, and Drosophila melanogaster couldn't make it that high. And you see their ranges are like this, so they don't go this north. So they couldn't come with the first humans coming to the north, the Americas, but they came with the colonialists when they were passing the Atlantic with their ships. Okay. The second thing we know is they can move. Like they move long distances. In late 70s and early 80s, Coyne has done some experiments. In the first one, they released some flies in Death Valley, and the flies moved very long distances, like I think 10 miles a day or something. And it was criticized very harshly, because people said the flies wouldn't fly in a normal environment. If you leave them in Death Valley, of course they will be moving, but if you would leave them with food, they wouldn't move at all. It was a big discussion back in the day. It's kind of it's still kind of a discussion, because there are very interesting things going on. And then Coyne did this experiment where they released flies in a Maryland orchard with a lot of food, and they started tracking these flies from that point and outside. In 3 months' time, they collected flies from 10 kilometers like distance, which is very, very interesting. This shows that the flies move, and they move. They're not really. They don't really stay at one point. They just move long distances. And they tracked these by, like, these flies were genetically modified flies, so they had genetic markers in them. They collected all the flies they could find, and then they ran gels and found, like, they tried to find their own flies in these gels. And the last thing we know is they show differences along the latitude. So the question kind of is this population different than this population or this population? And we can see there are clients, it's not really important what traits are changing, but this is a gene, couch potato. It changes along the cline, and this in this cline you see the change in the wing size. It's kind of, it follows a pattern. So there are clinal changes in the trait values. So this is one thing we have to know. And for the last thing we need to know is these are the genes from flies between spring and fall, and we see around like 0.2 change in their allele frequency from one season to the other, which is almost like, almost all the time like reproducible, like it just repeats. So there is some fluctuation of the allele frequencies, which is also again really cool, because 0.2 change in one season is a really high selection. They go through 10 generations per season. It's the selection coefficient here is really high. So what did I do? So I created three outbred populations, as I described before, from Maine, Pennsylvania, and Florida, using 100 isofemale lines each. And I tried to answer these questions. The first question is, like, I think the umbrella question for this is, what's the effect of migration on adaptation? And then we can break down this into like sub questions. The first one is, does migration have any effect at all? Because it might not have any effect. But if it does have any effect, does it increase or decrease the rate of adaptation? And finally, is the source of migration important? So again, reminding you that it's not really important, that these questions by themselves are not really important, might not be really important for you, or like, for the test purposes too. But you try to understand how I will address these questions at the end. So I created these populations, these outbred populations. I will use these as my control groups, but I need also some experimental groups. My experimental groups are Maine into Pennsylvania and Florida into Pennsylvania. I consider Pennsylvania as my resident population. So I'm saying I have a resident population here. It is Pennsylvania. What if I bring flies from Maine, which are, we can say, cold adapted, and flies from Florida, which we might say they are warm adapted? So what I did was I put a bunch of flies into some cages from Pennsylvania outbred population, and then brought some individuals from Maine into Pennsylvania, and then Florida into Pennsylvania. While doing this, I created 7 replicates of each of these, and I use these 3-to-1 ratios. But what will I compare my results to, right? So I have before and after, but I need some other thing that I can compare after to after. So what I did was I created a summer treatment, because the flies, like a season for a fly, we can say it starts around around June, and it ends around in like November. So these are the last days of flies around, if you go out and collect them. So we have 2 types of selection on these populations. One of them is summer selection, one of them is fall selection. So I created these environments in the lab, and I created 2 replicates of these 35 populations, put one into summer treatment and one into fall treatment. So I started with 35. At this point I have 70 cages that I'm taking care of. So, but I want to, like, by the way, I let them go for 5 generations, non-overlapping. Non-overlapping means I collected eggs, let them develop, but every time I collect eggs, I discarded the adults, so that there's no overlapping between the generations. So after the experiment, I want to compare these results. I want these results to be comparable to each other. What did I do? I put them in a common garden when they are finished for 2 generations, which is 25 degrees day and night, and like that's kind of the standard procedure. But the reason I'm doing this is I'm trying to get rid of any kind of epigenetic or maternal effects that are present in these groups. And then I measured their phenotypes. Okay, the first question I want to answer is, did my experiment work? So after I put these are measurements just after I put them together, and we see Pennsylvania, Maine, and Florida are completely different different places. So this means that they're different populations. We have to consider this plot as a space. So in the space they have different places. And PA Florida is closer to PA, and in between PA and Florida. This is good. This is what I wanted to achieve. Also PA Maine, it's closer to PA, and it's between PA and Maine. This result shows that the migration part of my experiment worked. Okay, the second question. Did the selection work? So to understand that, this is again a complex space. We have to look at this as, you know, where in the space these groups are. So this green space is before any kind of selection, and this blue space is after warm selection, and this red space is after cold selection. The blue space is after cold selection, and red space is after warm selection. So in this graph, what we see is the selection moves the population in a way that it is reproducible. So this is again a good result. This says that my selection worked pretty good. So like we answered 2 of the questions. Did selection work? Yes, it worked. Did my migration work? It worked. So like both of the most important parts we can answer yes, they work. So it's not okay. The results are not very important again. But what we see here is Um, this was before the selection. These, like, here this is a little bit more complicated. The red one is Florida, the green one is PA, orange one is PA Florida, and these round ones are before, the square ones are cold selection, and the triangles are warm selection. So this is basically the same thing we see. I just created some subsets. But what we see here is after cold selection, PA Florida, and Florida kind of end up in the same phenotypic space, but they slightly diverge in the warm selection. When we look at this one, we see PA main and main slightly diverges under cold selection, which is different from this one, but they slightly converge into the same space compared to their first starting points after cold selection, after warm selection, which is kind of an interesting result, because this shows the source of the migration might be really important. Okay, if we go into a little bit into details, what's happening if you look at the like a phenotype, like specifically. So this is what happens to larval development time throughout the season. This is from another experiment. This is not from my experiment, but this shows what's happening. So throughout the season, we are expecting the development time to decrease over time during the summer, and it should increase throughout the winter, throughout the fall. So do we see that? So if we look at the warm selection, the summer selection, we see all of them decrease, and if you look at the cold selection, we see all of them increase. Yes, they show the same, like the similar patterns we see in other experiments here. But what's more important here, what we see here is, like, we see PA Florida. We expect PA Florida to be in between PA and Florida, but it's not. It is a little bit down here. And again, we expect PA Maine to be here, like in between PA and Maine, but it's again not in between. And if you look at the cold selection, it happens here too. So this shows that migration might be adaptive too. Finally, what we did was, like all of the experiments I showed was in the lab, right? So now we want to know if our experiments are ecologically relevant. So to understand that, we did the experiment in an outside setting. I showed the experimental orchard before. We have a peach tree inside a cage and ground cover. So what we did was PA Maine and PA Florida, because Maine and Florida would be like irrelevant for this experiment. So we just did the summer season, 45 days overlapping generations, and we collected the samples. I will just show one single result. That is that the patterns we end up with after these indoor and outdoor experiments look similar, which means that our results, our selection patterns, were relevant with the summer selection, and we got similar results, which kind of shows that our most of the selection on larval development time is driven by the temperature patterns. And also our results are ecologically relevant. So to summarize, here I try to show you how I found some questions which are really important for me, and then using those questions I created some experiments to address those questions. And the steps I've taken to address all of the questions I did. So I want you to concentrate on that part. How do we solve if we are given a problem, or if we found there is an open question in science, how we solve that? I think this is it from my discussion. I will just check one thing. Let's see. Okay, 1 second. I will check one thing and then we will continue. I think I will end this. Let me see. Yeah, I think that's it. I was not sure if I addressed everything, but it looks like I addressed everything from the lecture. All right. See you soon.